Okay, so I haven't read through this paper yet. It's molecular mechanism for antibody dependent enhancement of coronavirus entry. Um, so I have not gone over this, and I'm surprised. I'm actually very surprised that I hadn't found this paper earlier, especially since one of the places or the locations that was contributing research for this paper was the Wuhan Institute of Virology at the Chinese Academy of Sciences in Wuhan. So that is very interesting, and it uh, going through this, this particular paper was received at this. It was received for or received by the Journal of Virology in November 27th, which is about the time that this virus, that the SARS coronavirus or SARS-CoV-2 ended up starting its, I would say, the where it became more apparent that person-to-person travel, or how do I phrase this? So basically, that would be about the time where the coronavirus started crossing into humans. Or if maybe it started crossing into humans far earlier, but that was about the time where human to human contagion was uh, much more apparent, or when it would have started doing human to human transmission. So I just find this extremely creepy because this paper was published, or not published, but submitted and received late November. So I'm unaware of how aware the researchers were and how important this type of research would have been at this particular point in time, but it is obviously something that either they had absolutely no clue and it was luck, or they did have a clue and they wanted to get this information out as quickly as possible. So either one of those cases, uh, I believe, I do. Part of me does believe that there might have been whispers going around by late November that there was coronavirus, a novel coronavirus epidemic going on in Wuhan at that time, late November, and so it is possible that the researchers at the Wuhan Institute of Virology tried to um, basically get a paper out, or a paper regarding this particular research out as quickly as possible. And I mean, what is surprising to me was that it was accepted the 11th of December. So this was actually quite an old, not old, but this is not new. This isn't breaking. This was out for a while, and I'm really surprised that I haven't heard any. I haven't watched any videos of people going over this particular paper, and I haven't seen in the news anything regarding this particular paper. So I'm really surprised that I was even stumbled upon it, to be frank. So what I'm going to do is I'm going to give just a quick synopsis, and I haven't gone over this paper. I'm actually going to read through it with you, and I'm not going to understand everything the first time through, but I do want to go over um, what is antibody dependent enhancement. It's not very well understood. We just do know that it is a problem for making diseases worse the second time you're infected. So you might have heard in the news a lot, like what are the reinfection rates, or what's the likelihood that you're going to get reinfected with this virus? That's a moot point, or I should say that is not proper semantics, because it's very likely, or I shouldn't say very likely, it is more likely than not that you will get reinfected with any disease. Any disease, you are most likely going to, if it's widespread, you're going to get infected with it multiple times. Okay, but the whole point of your immune system is that your immune cells and your antibodies sequester that infection before it develops a pathology the second and third time around, right? Um, but what ends up happening is over time, for all diseases, or I should say for all acute diseases that you get out of your system, usually your antibody concentrations go down, and they're supposed to go down. But the white blood cells that are able to produce those antibodies, those specific antibodies, they stay around, and when you do end up getting infected again, those white blood cells end up multiplying and producing a lot of antibodies, which ramp up the antibody concentrations for that specific disease the second time you get infected. Or maybe it's not the same disease, maybe it's a very closely related disease. Either way, if your antibody concentrations will go down. How far they go down, that depends on what disease it is. And we are, for coronaviruses in general, it's sounding like antibody titers do end up going down pretty quickly, which is what allows people to get infected with the common cold coronaviruses very, very quickly, within a few years, really. But that doesn't mean that the antibodies are worthless, because they're still protecting you. They're still doing their function to make sure that you don't die. And what we would hope is that when you do get infected with SARS-CoV-2 a second time, that the antibodies that you have produced and the white blood cells that produce those antibodies will be in high enough concentrations to prevent you from receiving serious illness the second time around. Okay, that is when things work out correctly. I forget the exact terminology, but I'll just call it, you can have fuzzy immunity and you can have hard immunity, where hard immunity is the antibodies and the white blood cells that you have are effective and you don't get any disease the second time around or third time around, X times around. And then there's a fuzzy immunity. I will look up the actual correct terminology later, but where you do get infected, but since you have those antibodies flowing in you, you are not going to be nearly as sick as the first time around. That would be a fuzzy immunity. But there is I guess you could argue that there's a third and fourth option. The third option is that you get just as sick every single time, which would mean that the antibodies that you produce are not enough, and every single time you get infected, you are just as sick every single time after the fact. That is the third option. But there is a fourth option, which is far worse, which would be the second time you get an infection, the infection is far worse than the first time you catch that infection. And specifically, this is called antibody-dependent enhancement, which is when the antibodies, if they get below a certain concentration in the body, and we do know that antibodies for other coronaviruses, their concentrations do go down very rapidly. If they do end up going down significantly, then they are no longer neutralizing. And then somehow, some viruses are able to take advantage of those low levels of antibodies in order to get close to immune cells. So if you could imagine, I'll just say the second time you get a virus and there's very few antibodies, those antibodies can stick to the coronavirus, and then but not be enough to completely shut it down. And in those particular cases, you can end up where the antibody will actually help recruit immune cells to be near it, or become close to the virus. And that would be another possible mechanism by which a virus could shuttle itself into a white blood cell and then allow them to duplicate very rapidly. And on top of that, I assume that it would. Okay, I'm not going to make too much assumptions, but so that would be a concern with antibody-dependent enhancement. So, do we believe that it might be the case for SARS-CoV-2 that antibody-dependent enhancement is playing a role? There is some belief that, or some, what would you call it? Some evidence to verify that this strategy is being employed, because when we were trying to develop vaccines for SARS-CoV or the original SARS and MERS, some of those vaccine trials had to be halted because in some animal models, the vaccines ended up making the disease far worse than without the vaccine because the vaccine was not providing sufficient antibody. It wasn't creating sufficient neutralizing antibodies. So, this is why a lot of people are concerned about this particular strategy being employed by SARS-CoV-2, because perhaps maybe a lot of the people who ended up being hospitalized, that might have been the second time they got infected with the coronavirus. It's very hard to, I guess, remove all of the convoluting factors, especially when the pandemic is in its early stages. But this is one of my concerns. So what I want to do is I'm going to go through this paper with you, all 4 of my subscribers, and try to make sense of this. And it's a somewhat long paper, so this might be a pretty significantly long video, but I just want to get it out because I want to learn this with y'all. So let's jump into it. So antibody-dependent enhancement, or ADE, of viral entry has been a major concern for epidemiology, vaccine development, and antibody-based drug therapy. However, the molecular mechanism behind ADE is still elusive. Coronavirus spike protein mediates viral entry into cells by first binding to a receptor on the host cell surface and then fusing viral and host membranes. In this study, we investigated how a neutralizing monoclonal antibody, or MAB, which targets the receptor binding domain of MERS, coronavirus spike, and remember this came out before SARS-CoV-2 was known about, or widely known about. So they're doing the research on MERS instead. It mediates viral entry by using pseudovirus entry and which target the receptor binding domain of Middle East respiratory syndrome, coronavirus spike, mediates viral entry using pseudovirus entry and biochemical assays. So they can use pseudoviruses, or I don't want to say fake viruses, but genetically engineered viruses that are missing a lot of the pathogenicity because they've removed a lot of the information within it, or a lot of the genetic material. They rendered a lot of the genes useless, and then they can replace some of those genes with genes from other viruses, and in this particular case they're adding the spike protein to that virus to do research on that spike protein. Our results showed that the monoclonal antibody, and I should say the monoclonal antibody would mean that they have one type of antibody that's attacking one portion of the spike protein, in contrast to a polyclonal antibody where they are using a lot of different antibodies from different white blood cells to basically attack many parts of a spike or any type of protein. And those would those would be more difficult to do studies on because you can't really uh, control concentrations nearly as easily when you're dealing with a lot of different types of antibodies. But regardless, continuing, allowing it to undergo conformational changes and become prone to proteolytic activation. So here, oh, okay, so here, oh, this is another technique. So they're showing that this particular antibody, if it binds to the virus surface spike, it makes so if this is the antibody and if this is the spike protein that is on the virus, and remember they're actually trimers of the spike protein, but if it binds, it actually changes the shape of the spike protein, which then makes it more susceptible to proteolytic cleavage. So a protease would come along and do a cleavage of the spike, which is those cleavages are usually necessary for the virus to end up infecting cells. Interesting. So allowing it to undergo conformational changes to become prone to proteolytic activation. Meanwhile, monoclonal antibodies bind to cell surface IgG, Fc receptor. Oh, okay. Guiding viral entry through a canonical viral receptor dependent pathway. So this is a receptor on a certain type of white blood cell. Let me look this up just to make sure. So this is the Fc receptor on the NK natural killer. I think, yeah, NK cells. So these are a type of immune cells that you have in your body, and they recognize so if there's an antibody that's sticking onto something, these natural killer cells identify that area on the antibody, which tells them what to consume. So here you can see a Fc receptor is a protein found on the surface of certain cells, including among others B lymphocytes, follicular dendritic cells, and second of the B cell areas of the lymphoid tissue. Yeah, so natural killer cells, macrophages, eosinophils, basophils, human platelets, and mast cells. What were those again? Is a migrant cell of connective tissue that contains many granules rich in histamine and heparin. Really? So histamine is one of the, if you take antihistamines, it ends up helping you suppress allergic responses. And then heparin, which is one of the iron metabolism hormones. So interesting. This to me, oh, and it's also part of the immune and neuroimmune system. So I gotcha. So these are all immune cells, right? And here you can see the Fc receptor on whatever cell we're talking about. And here's an antibody that is connected to a pathogen. So in an ideal world, this antibody connects to the pathogen, and then the effector T cell, or the white blood cell, or the immune cell, would recognize the antibody, and it would tell the white blood cell to consume the pathogen to help sequester it. Okay? So that is the Fc receptor, or the IgG Fc receptor, because these are specifically IgG antibodies, which are more long-lived than the IgM antibodies. Continuing, this guides viral entry through canonical viral receptor-dependent pathways. So basically, rather than being eaten, which is what the white blood cell wants to do, the viral receptor is now actually making the virus more able to infect the white blood cell. which is what I said earlier. Our data suggests that the antibody FC receptor complex functionally mimics viral receptor in mediating viral entry. So normally, with the SARS-2 coronavirus, usually utilizes the ACE2 receptor. And the ACE2 receptor is on certain cells of your body, and then the spike protein of the virus interacts with that ACE2 receptor. And then later, a protease will come along and do a cleavage of the spike protein, which then allows the virus to enter the cells. Okay, but instead of that process, these these virus particles are getting funneled into the white blood cells through the FC receptor instead of the ACE2 receptor. So this would indicate to you that these particular viruses are able to use multiple modes of entry. Okay, so what does this tell me? Well, this tells me that since they are utilizing multiple strategies of getting into cells, then that would tell me that this virus is probably more suited at crossing species barriers. Because, especially if it's utilizing antibodies to do that, well, if the process is independent of the actual shape of the receptors, or I should say of the normal receptors, and is instead utilizing antibodies, which are going to be tailor-made for whatever spike protein you have, then that means that for a lot of creatures, their immune system might actually permit the vulnerability, or I should say, they are going to permit being infected by these viruses the second time around. So maybe the first time around, the virus doesn't really do much. They just make a few antibodies, which gets it out of solution. Fine, it'd be like pollen. You make antibodies against pollen if you breathe it in. So that could happen to a different animal. But then the second time around, when they get infected, even though the spike protein isn't there, or even though the ACE2 receptor isn't present in that other host, they're not even going to be using that method of getting into the cell. They might just be using the antibodies to get into the cells and start the replication. So this would be a strategy to be able to infect multiple species, which we do see with with SARS-CoV-2 at least. And some other coronaviruses are also pretty adept at crossing species barriers. One of the human coronaviruses crossed recently from the bovine, or from cows, or cattle, I should say. And not just cattle, but a lot of different split-hoofed creatures are susceptible to the bovine coronavirus. Interesting. So let's continue. It functionally mimics viral receptor in mediating viral entry. Moreover, we characterized monoclonal antibody dosages in viral receptor-dependent, FC receptor-dependent, and both receptor-dependent viral entry pathways. So they're seeing how the monoclonal antibodies will affect the entry of the virus with just the receptor of choice and with just the FC receptor, which is the antibody IgG receptor, and both. And remember, this is being done on MERS, not on SARS-CoV-2. So MERS, I believe, uses a completely different receptor to get into the cells, at least in humans and in dromedary camels. Continuing, our study reveals a novel molecular mechanism for antibody-enhanced viral entry and can guide future vaccination and antiviral strategies. So what's the importance? Antibody-dependent enhancement of viral entry has been observed for many viruses. It was shown that antibodies target one serotype of viruses, but only subneutralize another. So if you make antibodies for one type of virus, maybe a different virus that is a closely related strain is able to be subneutralized by the antibodies. And by subneutralized, I mean a neutralized virus would not be able to infect a cell because there are too many antibodies surrounding it. But if it's subneutralized, then the virus is still infectious, and then it has a few antibodies on it that direct it directly to a white blood cell that has now become susceptible to that virus when it normally wasn't. Let's continue. It was shown that okay, here we identify a novel mechanism for ADE. A neutralizing antibody binds to the surface spike protein of coronavirus like a viral receptor, triggers a conformational change of the spike, and mediates viral entry into IgG Fc receptor expressing cells. So I assume they are going to genetically engineer some cells to have this Fc receptor, which would normally just be on, or predominantly immune cells, but they're going to put it onto a different cell that doesn't normally have these receptors. And anyways, through canonical viral receptor-dependent pathways. We further evaluated how antibody dosages impacted viral entry into cells, expressing viral receptor, Fc receptor, or both receptors. This study reveals complex roles of antibodies in viral entry and can guide future vaccine design and antibody-based drug therapy. So one indicator that antibody-dependent enhancement is not an issue with the SARS-CoV-2 coronavirus is that by taking plasma from recently infected or recently recovered patients and injecting that into people who are still infected, that seems to be benefiting the people who are still infected. But this is utilizing people who probably have a high concentration of antibodies in their body. So, and also they're probably dealing with very similar strains of coronavirus. This does not say that 3 months down the road that those survivors are going to have useful concentrations of antibodies in their system anymore. They might have become subneutralizing at that point. And then also, this doesn't say anything about if the virus does some key mutations which change the shape of their spike significantly. Maybe those exact same antibodies are going to have the opposite effect, where they are no longer neutralizing, but then they are subneutralizing later. We haven't done those experiments yet, but hopefully, I mean, this is one of my worries. Hopefully, that is not the case. But as far as how other coronaviruses behave, I'm not very confident. Anyways, let's go down. So here we have this DPP4, and this is the MERS receptor binding domain. So this is a part of the MERS coronavirus spike protein, and this is a portion of it that has the receptor binding domain. Okay, so let's read through the structural similarity between DPP4 and monoclonal antibody in binding MERS-CoV spike. A is the tertiary structure of MERS-CoV receptor binding domain in complex with DPP4, which I believe is the receptor. It's peptidase. Actually, let's look this up right now. Yeah, dipeptidyl peptidase 4 in dipeptidyl peptidase 4. And I don't think it matters that much, but and this is the drug called some inhibitors of this dipeptidyl peptidase are, they're drug targets, I should say. But just know that this is a receptor on some of your cells, and MERS binds to it, which is what allows the MERS to enter the cell. So complex with that, and then the DPP4 is colored yellow, the receptor binding domain is colored cyan, and red is the receptor binding motif. So this entire thing is the receptor binding domain, but the red part is known as the receptor binding motif, which is explicitly, so this whole thing plays a role in binding, but this receptor binding motif has the role of the actual amino acids that are touching the dipeptidyl peptidase 4 receptor. So B, the modeled structure of MERS-CoV spike in complex with DPP4. So this would be the, and I believe E would mean the ectodomain, but do not quote me on that. So this would be the entire spike protein that is in complex with B. And remember, these spike proteins are trimers. So this is one of the trimers that is colored, and then one part of the spike protein that is colored, and then there are two others here that are not colored. And as you can see, out of the 3 spike proteins, only one of them is necessary to do the binding with the DPP4. And this is slightly tangential, but in a lot of the 4 human coronaviruses that are a lot less lethal, all of these, all 3 in this trimer are in the closed position. So they're all closed, or all pointing down. As you can see here, these 2 are pointing down. But in the SARS-CoV-2 and MERS, they have always been able to find a portion of the spike proteins that are partially opened. And in this case, you can see it is partially opened, especially when it's already binding. So if it's binding, it has to be open in some way. So continuing. Yeah, so one monomeric subunit is colored blue, and the other 2 are colored gray. To generate the structural model of the spike, I'm going to say ectodomain, so the outer part of the spike, in complex with DPP4, the receptor binding domain in panel A was structurally aligned with the standing up RBD of the SE trimer. So this is the one that's standing up, so that's why they're using it. And tertiary structure of MERS-CoV receptor binding domain, and critical monoclonal antibody binding residues were identified through mutagenesis studies and are shown as green sticks. So they did some studies on a monoclonal antibody. So they took a type of antibody that was known to bind to this active form of the spike protein, or of the receptor binding motif, and they identified some key residues on the receptor binding motif that interacted with the monoclonal antibody. Interesting. Anyways, let's keep on going down. Let's see. So it has been shown that when patients are infected by one serotype of dengue virus, i.e. the primary infection, they produce neutralizing antibodies targeting the same serotype of the virus. However, if they are later infected by another serotype of dengue virus, and serotype just means just the, what would be the best way to describe it? You know, I'll just get a, a serologically distinguishable strain of a microorganism. So this has very little to say about the genetics of the virus. It has more to say about the shape of the uh, extraviral proteins surrounding it. So sometimes you can do a few very select mutations. And so the genome, the genetic material of the virus is very similar. But if you can change just a few residues that can alter the shape of the protein significantly, that would be considered a different serotype because the antibodies attacking one of those spikes are going to be far inferior at attacking the coronavirus spikes of the other serotype. So, yeah. Yeah, these microorganisms, viruses, or cells are classified together based on their cell surface antigens, allowing the epidemiologic classification of organisms to the subspecies level. So keep in mind, these are viruses, these are pathogens that are still within the same species, but they present themselves in a completely different way. So another example that I can give is, let's say the coronavirus spike has a proteolytically active site on it, which makes it very easy to be munched up by a protease in your cell. So let's say it has one mutation where the spike protein is complete and whole, right? And so that's going to have a very specific shape when it is expressed and made and released into the environment. But what if there's a mutation which removes that proteolytic site? Well, now the entire that is going to have a completely different structure, and therefore would be very likely to be a completely different serotype. So that would be an example of how just one or two amino acid changes could completely change the antigenicity of the virus proteins. So continuing, however, if they are later infected by another serotype of dengue virus, the pre-existing antibodies cannot fully neutralize the virus. Instead, the antibodies first bind to the virus and then bind to the IgG-Fc receptors on immune cells and mediate viral entry into these cells. A similar mechanism has been observed for HIV and Ebola viruses. Thus, subneutralizing antibodies, or non-neutralizing antibodies in some cases, are responsible for antibody-dependent enhancement of these viruses. Given the critical roles of antibodies in host immunity, ADE causes serious concerns in epidemiology, vaccine design, and antibody-dependent drug therapy. This study reveals a novel mechanism for ADE in which fully neutralizing antibodies mimic the function of viral receptor in mediating viral entry into Fc receptor expressing cells. So coronaviruses are a family of large positive stranded and enveloped RNA viruses. Two highly pathogenic coronaviruses, SARS and MERS, cause lethal infection in humans. And wow, it just takes me back to read a paper that talks about coronaviruses, yet came just before SARS-CoV-2 became a thing. It truly is quite humbling. An enveloped anchored spike protein guides coronavirus entry into host cells. As a homotrimer, so a trimer with 3 of the same type of monomer, or subunits, I shouldn't say subunits, yeah, I'll just say monomers. The spike contains 3 receptor binding S1 subunits and a trimeric membrane fusion S2 stock. This state of the spike on the mature virions is called prefusion. SARS-CoV-2 and MERS-CoV, SARS-CoV and MERS-CoV recognize angiotensin converting enzyme and dipeptidyl peptidase 4, respectively, as their viral receptors. Their S1 subunits each contain a receptor binding domain that mediates receptor recognition The RBD, or the receptor binding domain, is located on the tip of the spike trimer and is present in two different states, standing up and lying down. So as you can see in this 3D structure here, this colored one is standing up. The receptor binding domain is exposed. And here, in these other two in the trimer, they are pointed down. Now, why would a virus want to have them pointed down? Well, if you generate an antibody uh, against this site, that might not necessarily be good for the virus to survive much longer. So a strategy is have your receptor binding domain be closed and hidden away. That way it's more difficult to generate antibodies against that particular domain. Binding to a viral receptor can stabilize the receptor binding domain in the standing up state. Receptor binding also triggers the spike to undergo further conformational changes, allowing host proteases to cleave at two sites sequentially, first at the S1-S2 boundary and then within the S2, i.e. the S2' site. Proteolysis of the spike can take place during viral maturation by proprotein convertases. Oh yeah, so for different viruses, I did another video on this. Sometimes the virus might even be cleaved by proteases even before the virus has even been fully formed. And so as the virus is forming, sometimes the virus can decide, oh yeah, cleave me before I'm even done. Or sometimes it will rely on the strategy to be cleaved only after it has attached to its victim cell later. There are many mechanisms by which viruses can basically just do a few mutations and they can regulate when these events are taking place, and that might be able to confer an advantage to the virus because they're able to mutate or change their serotype very, very quickly by just doing one or two mutations. Or after viral endocytosis by the lysosomal proteases. So sometimes the viruses, after they even get into the cell, after they've been sucked in by the cell, that is when they do the final protease activity. And oftentimes that is a way to be released into the cytoplasm because after the virus gets sucked in by endocytosis, it still has a membrane that it needs to exit. There's still a membrane preventing the virus from being in the cytoplasm. So there needs to be some other event, such as proteolysis, that breaks the bubble that it is inside of. Anyways, after two protease cleavages, S1 dissociates and S2 undergoes a dramatic structural change to fuse host and viral membranes. This membrane fusion state of the spike is called postfusion. Due to the recent progress towards understanding the receptor recognition and membrane fusion mechanisms of coronavirus spikes, coronaviruses represent an excellent model system for investigating ADE of viral entry. ADE has been observed for coronaviruses. Several studies have shown that the sera induced by SARS-CoV spike enhances viral entry into Fc receptor expressing cells. Further, one study demonstrated that unlike receptor-dependent viral entry, serum-dependent SARS-CoV entry does not go through the endosome pathway. Let me just reread that. Further, one study demonstrates that unlike receptor-dependent viral entry, serum-dependent SARS-CoV entry does not go through the endosome pathway. What is the endosome pathway? I forgot. The endosome is a membrane-bound compartment inside. It is an organelle of the endocytotic membrane, or endocytic membrane transport of okay. Interesting. I'll have to look into that later. Additionally, it has long been known that immunization of cats with feline coronavirus spikes leads to worsened future infection due to the induction of infection-enhancing antibodies. However, detailed molecular mechanisms of ADE of coronavirus entry are still unknown. We previously discovered a monoclonal antibody, or MAB, named MERS-MAB1, which has strong binding affinity for MERS-CoV receptor binding domain, and efficiently neutralizes MERS-CoV entry by outcompeting the DPP4. So this particular monoclonal antibody is so effective at binding to the SARS-2 coronavirus spike that it outcompetes the binding to the dipeptidyl peptidase 4 receptor, and therefore it makes it easier to sequester these MERS viruses. One second. This discovery allowed us to comparatively study the molecular mechanisms for antibody-dependent enhancement and receptor-dependent viral entries. In the study, we examined how MERS-MAB1 binds to MERS-CoV spike, triggers the spike to undergo conformational changes, and mediates viral entry into Fc receptor-expressing cells. We also investigated the pathway and antibody dosages for MERS-MAB1 dependent and DPP4 dependent viral entries. Our study sheds light on the mechanisms of ADE and provides insight into vaccine design and antibody-based antiviral drug therapy. So here are the results. So antibody-dependent enhancement of coronavirus entry. To investigate ADE of coronavirus entry, we first characterized the interactions between MERS-MAB1, remember this is the antibody, the monoclonal antibody, which is a MERS-CoV receptor-specific monoclonal antibody, and MERS-CoV spike using biochemical methods. First, enzyme-linked immunosorbent assay, or ELISA, was performed between MERS-MAB1 and MERS-CoV receptor binding domain between MERS-MAB1 and MERS-CoV spike ectodomain. To this end, MERS-MAB1, which was in excess, was used to coat the ELISA plate, and gradient amounts of recombinant RBD, receptor binding domain, or the spike ectodomain, were added for detection of potential binding to MERS-MAB1. The results showed that both the receptor binding domain and the spike ectodomain bound to MERS-MAB1. So the spike ectodomain bound to MERS-MAB1 more tightly than just the receptor binding domain, likely due to the multivalent effects associated with the trimeric state of the spike ectodomain. So sometimes these receptor binding domains, they work by themselves, but they don't exist in a vacuum. They normally exist connected to the rest of the spike protein. So an effective evolution strategy would be to be more effective as the whole spike rather than just being the one part that is that has those amino acids on them. Continuing, second, we prepared FAB from, and this is the fragment. So they prepared the fragments from MERS-MAB1 using papain, which is a protease from the papaya digestion. So rather than doing research on the entire antibody, they did research on just the fragment that does the binding, and examined the binding between FAB and the spike ectodomain using ELISA. Recombinant spike ectodomain was used to coat the ELISA plate, and gradient amounts of FAB or MERS-MAB1 were added for detection of potential binding to the spike ectodomain. The results showed that both the FAB and the MERS-MAB1 bound to the spike ectodomain. MERS-MAB1 bound to the spike ectodomain more tightly than just the fragment did. So remember, the FAB is just a fragment of the antibody. Also likely due to multivalent effects associated with the dimeric state of MERS-MAB1. So remember, antibodies aren't just they're not just the fragment that binds to its victim. They are an entire structure, and that entire structure usually plays a positive role in binding to the antigen or to the epitope. Third, a flow cytometry assay was carried out to detect the binding between the spike ectodomain and DPP4 receptor, and among spike ectodomain, mersmab1, and CD320A, which is which is the one of the FC receptors. So apparently there are multiple FC receptors, and this is just one of them. CD32A. Okay. To this end, DPP4 or CD32A was expressed on the surface of human HEK293T cells. Oh, and these are a culture of human kidney cells, and I'm guessing that's human epithelial of the kidney. So, just a quick background. So, biochemists and a lot of research likes to use, rather than doing experiments on full living organisms, which you need to care for and try to control for all the variations of their life cycle to have reproducible results. Oftentimes we like to do experiments on just a cell line, so just a culture of cells that exist, and you're sort of treating them like bacteria. You're treating them like a culture, right? And by doing this, you are able to control for a lot more variables, and it makes it easier for your research to be reproducible in different laboratories. And also, it's easier to do genetic engineering and modifications to those cell lines to see what happens when you add or subtract certain genes from them. And recombinant spike ectodomain was added for detection of potential binding to one of the two receptors. In the absence or presence of the mersmab antibody, the results showed that without mersmab1, the spike ectodomain bound to the DPP4 only. In the presence of mersmab, the spike ectodomain bound to the FC receptor, so the CD32A. As a negative control, a SARS-CoV receptor binding specific monoclonal antibody did not mediate the binding of the spike ectodomain to CD32A. Wait, as a negative control, did not mediate. Oh yeah, so if I will. Oh yeah. So they used. Sorry, I misread it for a second. So they used the monoclonal antibody, not of MERS, but they used a monoclonal antibody of SARS, which is not supposed to bind to the receptor binding domain of the MERS. So this is in contrast to MERSMAP1. And so they wanted to test whether or not just any antibody could be affecting the binding to the CD32A. And it turns out, no, that it doesn't do it. So that could tell you that it's not just any antibody that is binding to the receptor, binding to the FC receptors, and then allowing the virus to get in. It is explicitly the ones that are binding to the spike proteins. And here they're using ones that explicitly bind to the receptor binding domain, which theoretically could be making the virus much more prone to fusion. So maybe if we make an antibody, or if you produce an antibody that doesn't attack the receptor binding domain, maybe attacks a completely different domain, maybe you won't be seeing these types of antibody-dependent enhancement. That's a theory for now, but let's continue. And it did not mediate the binding of the spike ectodomain to the CD32A. The cell surface expression of both DPP4 and CD32A, which was remember this is the immune receptor and this is the normal receptor for MERS, were measured and used for calibrating the flow cytometry results, demonstrating that the direct binding of the spike ectodomain to DPP4 is stronger than the indirect binding of the spike ectodomain to CD32A. So normally, so this is saying that normally the spike does bind to its native or its usual receptor uh, much more so than the indirect antibody method to the CD32A. Granted, this is with this particular antibody. It doesn't say much about if you had a completely different antibody, which one would be better or worse. Overall, these biochemical results reveal that MERSMAB1 not only directly binds to the receptor binding domain region of MERS-CoV spike ectodomain, but also mediates the indirect binding interactions between MERS-CoV spike ectodomain and the Fc receptor. So next, we investigated whether or not MERSMAB1, which is the antibody, mediates the MERS-CoV entry into Fc receptor expressing cells. To this end, we performed, so now they're not just working on the spike protein. Now they're working on the full-fledged virus. To this end, we performed a MERS-CoV pseudovirus entry assay in which retroviruses pseudotyped with MERS-CoV spike were used to enter human cells expressing the CD32A on their surface. So oftentimes, we do not want to do research on the highly pathogenic virus in of themselves because it's dangerous to do that. And also, it's harder to do the genetic engineering experiments on them that we want to do. So oftentimes, we will work with a highly studied model virus or pseudovirus, and usually we removed a lot of the important viral components from them. That way, oftentimes, I don't know about if it's the case for this particular virus or pseudovirus, but it would usually make it so that if, for instance, if the virus did end up escaping from the lab, that virus would be incapable of multiplying outside of the lab environment. Oftentimes you can put these fail-safes in to make sure that the virus remains contained within the lab. And if you're working with the actual virus, it ends up becoming a little bit more or a lot more dangerous. And then also, if you are doing work on these highly studied pseudoviruses, it allows you to do, it gives you more reproducibility. It gives you, you're able to understand the entire virus more than the native virus, in which there are still a lot of things that are unknown about that new and emerging virus. You can do research on the pseudovirus instead, and then study the components of the emerging virus one by one. Anyways, the main advantage of a pseudovirus entry assay is to focus on the viral entry step, which is mediated by MERS-CoV spike, by separating viral entry from other steps of the viral infection cycle. Example, replication, packaging, and release. So this is another thing. Sometimes the viruses are able to go through different types of post-translational modifications before they are even in the final virus. So there's a lot of noise and there's a lot of variety at which a virus is able to change itself in its normal state, right? So if you wanted to learn something about one particular mechanism that the virus is using, you need to remove as much noise as possible. And that's pretty hard to do when you're dealing with a fully functioning virus, and viruses are known to be using multiple mechanisms and strategies to basically make it harder for them to be caught by an immune system, right? So by removing all of those other abilities and just focusing on one particular mechanism, you remove the noise and you make it easier to make a more definitive statement about what the virus is actually doing. Anyways, we tested 3 different types of the Fc receptors, so CD16A, CD32A, and CD64A, and each of these Fc receptors was exogenously expressed in the HEK293 T cells, which were the human kidney cells, if I remember correctly. Yeah, human kidney cells. We also tested macrophages in which mixtures of FC receptors were endogenously expressed. So just normally expressed in these white blood cells or macrophages, which are a type of white blood cell. The absence of the MERSMAB1 served as a control for MERSMAB1. A non-neutralizing monoclonal antibody would be appropriate as another control for MERSMAB1, but we do not have access to any non-neutralizing monoclonal antibody. So they wanted to use a different monoclonal antibody, one that wasn't completely neutralizing, and that would be a pretty effective that'd be a pretty effective control, but they didn't have access for a different monoclonal antibody against MERS. Unfortunately, that could have told us a lot more as well. The results showed that in the absence of MERSMAB1, MERS-CoV pseudoviruses, which remember this is the pseudovirus or the fake virus that has now been genetically engineered to express the MERS-CoV spike protein. It could not enter the FC receptor expressing cells. So without the antibody, the virus was not able to enter the FC receptor expressing cells. But in the presence of the antibody, the MERS-CoV pseudoviruses demonstrated significant efficiency in entering the CD32A expressing kidney cells and the macrophages. So it wasn't just the genetically engineered kidney cells that became vulnerable. The macrophages themselves also became vulnerable as well, and these also express the CD32A. In comparison, in the absence of MERSMAB1, which is the antibody, the MERS-CoV pseudovirus entered the DPP4 expressing kidney cells efficiently, but the entry was blocked effectively by MERSMAB1. So let me read this closely again. In comparison, in the absence of MERSMAB1, MERS-CoV pseudovirus entered the DPP4 expressing cells efficiently, but the entry was blocked effectively by MERSMAB1. Okay, so if they, oh, okay, I understand it. So these kidney cells did not express the FC receptor, right? They did not express the FC receptor. So if you add the antibodies, those antibodies are going to coat the virus. But those antibodies made the macrophages or the white blood cells susceptible, but it made the normal kidney cells it protected the normal kidney cells, and presumably it'd be because the kidney cells do not express the FC receptor. So it was able to protect the DPP4 receptor from entry, but it was not enough to protect the white blood cells from the FC mediated entry, and in fact, that's what allowed them to enter the FC cells, such as the white blood cells, the immune cells. In control experiments, anti-SARS monoclonal antibodies did not mediate the MERS-CoV pseudovirus entry into FC receptor binding kidney cells or macrophages. So they used a different antibody. So you could have used the MERSMAB1 antibody, which does do the antibody-dependent enhancement in the immune cells, but to test to see whether or not the just antibodies in general could mediate that fusion, they tested it with the anti-SARS, which is a completely different monoclonal antibody, and that did not end up working. So, and also those antibodies did not protect the normal kidney cells from being infected. So there was no cross-reactivity with this particular monoclonal antibody, but remember, these monoclonal antibodies are attacking the receptor binding domains, which is very specific and is there's a lot of variety between the receptor binding domains of these spike proteins. But there are some conserved areas of the spike proteins which, if you have an antibody that is able to attack those conserved regions, there is some belief that those antibodies are going to be cross-reactive, and they're going to be doing a protective effect on both of them, right? In another set of controlled experiments, we showed that neither the Fc receptor nor the fragment portion of MERS-MAB1 could mediate MERS-CoV pseudovirus entry into the Fc receptor expressing kidney cells nor the macrophages. So this is telling you that you need the entire antibody structure to do this antibody-dependent enhancement. So you cannot just have the FAB fragment, which still will bind to that receptor, or I shouldn't say it'll still bind to the spike receptor binding domain. It'll still bind to the virus, but since it is missing the parts that are useful for the antibody dependent enhancement, then they are going to be ineffective. They're going to be ineffective at playing a role in ADE. So, should I go over this one first? So this is the ELISA binding. Might as well. So this is the figure 2. This was stuff that we talked about a little bit earlier. But interactions between the coronavirus spike and the receptor binding domain of the monoclonal antibody. A is the ELISA for detection of the binding between the MERS receptor binding domain, i.e. MERS-MAB1. So the monoclonal antibody was coded to the plate in excess, and then if you have PBS, you have no binding, or you have your noisy binding, but it's pretty much nothing. And then if you give it the entire spike protein, you get a lot of binding, and then as you lower the concentration of it, it goes down, which makes sense. And if you just have the receptor binding domain, it also starts high and goes down. But one could look at this and say, okay, the receptor binding domain, if it's connected to the rest of the spike protein, then that allows it to bind a lot more effectively. So as you lower the concentrations, a lot of it ends up not binding if it doesn't have the rest of the spike. So that makes sense. And then let's go. Binding affinities were characterized by ELISA signal at optical density at 450 nanometers. PBS was used as negative control. ELISA for detection of the binding between FAB. Okay, so, and here they did the same experiment, but they did it with just the FAB, or the fragment of the monoclonal antibody, just to make sure that still worked as well. And here you can see, they wanted to see if it worked at all, and obviously it didn't. It worked, but it didn't work as well as a full monoclonal antibody. So here, the ELISA plate was coated with the spike protein of MERS, and this can tell you how much of the this tells you how much of the entire antibody would stick to the spike at the different concentrations, and this tells you how much of the fragment of the antibody would stick to the plate. And as you can see, it's not as effective, but it still does work at high concentrations. And this is telling you the binding, or fluorofax. This is the cytometry, right? Fax cytometry. So let me scroll through this. Flow cytometry for detection of the binding between MERS-CoV spike ectodomain and the DPP4 receptor, and among. Okay, so mock DPP4. One is when you have the MERS-CoV spike. Okay, 2 is when you have the MERS-CoV spike ectodomain added on these cells. Whew. Okay. So these are with the DPP4 expressing cells, and as you can see, if you add the MERS-CoV spike ectodomain, it then causes the sticking, which is why you're getting a higher signal. And if you, for 2, if you, oh, and this is when they're using the FC receptor, which is on the white blood cells. 2 is when you have the spike expressed, and the spike alone doesn't do anything, but if you have both the spike and the anti-MERS antibody, that allows the sticking to happen with the white blood cells, except it's not nearly as good as the native receptor. And 4, they tested it again, but they used the anti-SARS monoclonal antibody, and the anti-SARS monoclonal antibody did not allow for the sticking. Okay, but we already talked all about this, so I think we can skip the rest of that. So here at this figure, we have the ADE of coronavirus entry. This is all what we spoke about before. If you want to look at this yourself, I'll leave the description I will leave the link in the description so that you could look at this for yourself. But pretty much it's basically what we just said earlier, that there is antibody dependent enhancement, and it's done by these antibodies that are able to attack the receptor binding domain of the MERS coronavirus, and by doing so, it allows them to get into the I should say the FC receptor containing cells. So, and then there are different FC receptors. There's the CD16A, the CD32A, and the CD64A. And as you can see, for all of them, all of the ones that they have the receptor being expressed in them, all of them will show an increase of vulnerability to the MERS pseudovirus. And then the macrophages also have vulnerability to the pseudovirus, but only if you add the anti-MERS monoclonal antibody. Yeah, I already said all that. So let's say suggesting that both the FC and the FAB portions of anti-MERS monoclonal antibodies are required for antibody-mediated viral entry. The above mentioned DPP4 expressing HEK293 T cells were induced to exogenously express high levels of DPP4. To detect background expression levels of DPP4, we performed reverse transcription quantitative PCR, or QT-RT-PCR, on these kidney cells. The results showed that the kidney cells express very low levels of DPP4. In comparison, MRC5 cells, or human lung cells, express high levels of DPP4, where the HeLa cells, the human cervical cancer cells, do not express DPP4. Because of the comprehensive control experiments that were performed, the very low endogenous expression of DPP4 in the kidney cells should not affect our conclusions. Nevertheless, we confirmed the above results using HeLa cells that do not express DPP4. Overall, our results reveal that MERSMAB1 mediates MERS-CoV entry into the FC receptor expressing cells, but blocks the MERS-CoV entry into the DPP4 expressing cells. So that's basically what we said earlier. If they express the, actually let's see if we can look at it. So this is, I'd assume, yeah. So here they're looking at the HeLa cells as well. So they probably did the experiments on the kidney cells, which have a very low expression of the DPP4, and then they were exogenously induced to produce more DPP4, which would have made them vulnerable. But they also decided to use HeLa cells just to verify those findings, because HeLa cells apparently do not express any of the DPP4. And yeah, HeLa cells, no antibody, anti-SARS MAB, and anti-MERS MAB. The control, they express the DPP4. So if it's no antibody or if it's the wrong antibody, they get in. And if they have the anti-MERS antibody, they do not get in. But then if the HeLa cells are then expressing the CD32A, or the also known as the Fc receptor, then no antibody or the anti-SARS monoclonal antibodies, they're still protected. But if you add the anti-MERS monoclonal antibody, they then become susceptible. And they're still less susceptible than if they have the actual DPP4 or the normal receptor that binds to the receptor binding domain of the spike protein. But it's still something. The antibodies are making them more vulnerable. And if you think about this, your white blood cells are throughout your entire body, right? So if you have this antibody-dependent enhancement, it's going to be infecting a lot of cells throughout your entire body, which would be a more systemic disease. Whereas if the virus is only going to be infecting these DPP4 containing cells, well, maybe that's going to be in a smaller, that's only going to be infecting a few cell types, which could be very beneficial because then it's easier to contain wherever that disease is, or it's easier to target treatments for a single organ that is experiencing distress. But if it's your entire system that's experiencing distress, which is what would happen in antibody-dependent enhancement, then even if it's not nearly as effective on a per-cell basis, it'll be effective in that it's infecting a lot of cells throughout your entire body. Continuing, molecular mechanism for antibody-dependent enhancement of coronavirus entry. To understand the mechanism of ADE, we investigated whether MERS-MAP1 triggers any conformational changes of the MERS-CoV spike, and it was shown previously that DPP4 binds to the MERS-CoV spike and stabilizes the receptor binding domain in the standing up position. So remember, the trimers can be in the bound position or the closed position, or they could be in the standing up position. And the standing up position is where when the receptor binding domain is actually exposed and actually able to do its job. This resulting in a weakened spike structure and allowing the S2' site to become exposed to proteases. We repeated this experiment. MERS-CoV pseudoviruses were incubated with DPP4 and then subjected to trypsin cleavage. Okay. The results showed that during the viral packaging process, virus surface-anchored MERS-CoV spike molecules were cleaved at the S1, S2 site by proprotein convertases. In the absence of the DPP4, the spike molecule could not be cleaved further at the S2' site by trypsin. These data suggest that only the S1, S2 site and not the S2' site was accessible to proteases in the free form of the spike trimer. So the conformational changes of the receptor binding domain actually binding to the receptor is what allows the receptor to be proteolytically, or not the receptor, but what allows the spike protein to be attacked by a protease. And this presumably allows the virus to get into the cell, or into the cell more readily. In the presence of DPP4, a significant amount of MERS-CoV spike molecules were cleaved at the S2' site by trypsin, indicating that the DPP4 binding triggered a conformational change to MERS-CoV spike to expose the S2' site. Interestingly, we found that MERS-MAP1 binding was also allowed MERS-CoV spike to be cleaved at the S2' site by trypsin. As a negative control, the SARS-CoV receptor binding monoclonal antibody did not trigger the MERS-CoV spike to be cleaved at the S2 prime site by trypsin. Hence, like DPP4, MERS-MAB1, which is the antibody against MERS, triggers a conformational change of the MERS-CoV spike to expose the S2 prime site for proteolysis. So for this particular mechanism of the ADE, they're saying that the first part is the receptor binding domain needs to be bound to an antibody, or it needs to be in the partially open state. Okay. Okay, we'll keep on going. We further analyzed the binding between MERS-MAB1 and MERS-CoV spike ectodomain using the negative stain electron microscopy. We previously demonstrated through mutagenesis studies that MERS-MAB1 binds to the same receptor binding region of MERS-CoV receptor binding domain as the DPP4 does. So they did mutagenesis studies where they mutated the key amino acid residues that are known to allow the receptor binding domain of the spike protein to interact with the DPP4 receptor, and when they did the mutations that rendered it unable to bind to the DPP4, those same mutations also made the antibody not work either. Because full-length MERS-MAB1, which is a dimer, triggered aggregation of the spike ectodomain, which is a trimer, we prepared the fragment part, which is a monomer of MERS-MAB1, detected the binding between the fragment and the spike, and used FAB in the negative stain electron microscopy study. The results showed that FAB bound to the tip of the trimer, where the receptor binding domain is located. Due to the limited resolution of the negative stain electron microscopy, we could not clearly see the conformation of the FAB-bound receptor binding domain. However, based on previous studies, the receptor binding site of the RBD in the spike trimer is accessible only when the RBD is in the standing up position. Hence, the fact that the monoclonal antibody binds to the receptor binding region of the RBD in the spike trimer suggests that the RBD in the standing up state is in the standing up state. Thus, the result from negative stain electron microscopy and the proteolysis study are consistent with each other, supporting the idea that the DPP4, that like the receptor, the antibody stabilizes the receptor binding domain in the standing up position and triggers a conformational change of the spike. A future study of the high-resolution cryo-EM structures of MERS-CoV SE trimers, or a spike trimer complex with MERS-MAB1, will be needed to provide detailed structural information of the MERS-MAB triggered conformational changes of MERS-CoV spike ectodomain. Let's see how much longer this goes. I'm deciding whether or not if I want to go through the rest of this paper or just skip down to the discussion, because that stuff was not as important. So figure 5, pathways for antibody-dependent enhancements of coronavirus entry, impact of the pro-protein convertases. Okay, so this stuff might be getting a little bit too technical for me to explain properly. What I presume they're doing is they're doing a bunch of small experiments where they're toggling one variable and seeing which variables are important for the antibody-dependent enhancement of the coronavirus. So here, they pretty much established that the ADE is happening in MERS pretty well. And it's happening, they've only proved it when they're using a monoclonal antibody that binds the receptor binding domain. So here, I believe that they're justifying with other experiments, a possible mechanism for this to work. but I don't think that it'll be very fruitful for my listeners. If you want to read this yourself, I'll leave it in the description. But let's just go down to the discussion, where we will just uh, presume that everything they say is in agreement with the rest of the paper, and this paper was peer reviewed, so their discussion cannot vary wildly from the materials and methods. So discussion. ADE, a viral entry, has been observed and studied extensively in the flaviviruses, which are, a lot of them are mosquito-borne viruses, which is probably why that they were studied so much, particularly dengue virus. It has also been observed in HIV and Ebola viruses. For these viruses, it has been proposed that the primary viral infection of hosts led to production of antibodies that are subneutralizing or non-neutralizing for secondary viral infections. These antibodies cannot completely neutralize secondary viral infections, but instead guide virus particles to enter the Fc receptor expressing cells. Oh, so let me just look at this real quick, because they might have done a mutation study to see if the monoclonal antibodies actually made things worse if there was a mutation. Oh, well, something that should be noted. Um, they did do a study. They were also looking at the concentration of the monoclonal antibodies to see how those ended up affecting virality. And it looks like if the cell only expressed the DPP4, then as you increased the monoclonal antibody, less and less of the MERS pseudovirus was able to get into the cell. So as you increase the concentration, there is less and less that is able to get into the cell. These are the kidney cells, and with the kidney cells, if they express the DPP4, as you increase the monoclonal antibodies, less and less are able to get into the cell. But if they express the CD32A receptor, then as you add more monoclonal antibodies, you end up getting more and more that are able to enter the cell. But then as you add neutralizing amounts of the antibody, then the amount of MERS that are able to get in ends up decreasing. Right. So this could be important, knowing that for a lot of coronaviruses, your antibodies that you produce end up decreasing significantly within a few months, which is what allows some people to be infected by the same species of coronavirus multiple times throughout their lifetime. So what this can tell you is, let's say you do have high concentrations of antibodies that could protect you from having illness when you get infected a second time. But if you allow your virus titers or your virus amounts to get low, then you might, even if you are protected from the normal DPP4 entry, and this is for MERS, for SARS-CoV-2 and SARS, it would be the ACE2 receptor, not the DPP4. If you got infected again, maybe the amount of antibodies you have will protect your normal cells or your DPP4 cells from being infected, but they are simultaneously making your macrophages or your CD32A expressing cells vulnerable. So if you were in this particular concentration of antibodies in nanograms per milliliters, that might actually be not just as ineffective as having no antibodies at all, which, no antibodies at all, that's at 100%, right? But here you have to deal with, 30% of your normal cells or your ACE2 or your DPP4 expressing cells being vulnerable, and 50% or 40% of your CD32A cells or expressing cells being vulnerable. So you might just be, and remember, you might have a lot more white blood cells than you do your lung cells, right? So I shouldn't say lung cells, but you might be more vulnerable um, in other parts of your body if your white blood cells are getting infected. Whereas if it's just your lung that's getting infected, then that has its own set of problems, but that wouldn't be nearly as bad as having your immune system uh, start fighting against you. Um, oh, and here we go, showing if you have the HEK293 or the kidney cells that express both DPP4 and CD32A, it's like you're adding these two curves together, where as you increase antibodies, yeah, it's protecting your cells in these very low concentrations, but eventually as the concentrations get higher, you then start initiating this antibody-dependent enhancement of these cells. And so it goes up momentarily, and then hopefully, if you are expressing a lot of antibody, then it will be decreasing because then the antibodies will be neutralizing the virus rather than just mediating the antibody-dependent enhancement, right? And I believe it looks like they did the same thing with the MRC5 cells, where it goes down as you increase the concentration, and then boop goes back up at here it's at 500 nanograms per milliliter, and then goes back down when you get to very high concentrations of antibodies. And I don't know what the physiological concentrations of antibodies will be in your body, especially for the SARS-CoV-2 coronavirus. Just know that for MERS, this is happening. This is an event that is happening that you need to be concerned, and at least, if not concerned, at least aware of. You need to just be aware of your own vulnerabilities, is what I'm trying to get at. So yeah, maybe I should have gone over this, but these graphs have explained everything pretty perfectly. So continuing down, for these viruses, so for HIV, Ebola, and the dengue virus, for these viruses, it has been proposed that the primary viral infections of hosts led to production of antibodies that are subneutralizing or non-neutralizing for secondary viral infections. These antibodies cannot completely neutralize secondary viral infections, but instead guide virus particles to enter the Fc receptor expressing cells. ADE can lead to worsened symptoms in secondary viral infections, causing major concerns for epidemiology. ADE is also a major concern for vaccine design and antibody-based drug therapy. So an example would be when you make you can have antibodies injected into your own body from someone who has fully recovered from a disease. You can have their plasma injected into you if you are still suffering from the disease. And for SARS-CoV-2, it sounds like that is actually working effectively at getting people healthier and recovering more quickly. Since antibodies generated or used in these procedures may lead to ADE, ADE has been observed. Oh, and that they're saying that it's important to keep that in mind before you just start doing these experiments willy-nilly. Because let's say someone recovered from SARS-CoV-2 a year ago. Well, the antibodies that they produced might be at a low enough concentration where adding the antibody actually makes things worse, because they're not giving a concentration that's large enough to fully neutralize the viruses. Maybe they're giving enough to go into this antibody-dependent enhancement, and that would not be an effective treatment. Granted, one could argue that okay, this should also have an implication on as your body is producing antibodies in the first place. So as the virus is multiplying you during your first infection, it is possible that that you will go through a phase where the number of antibodies you produce is going to be at that small concentration because you're still ramping up your antibody production. During that time, it is possible that that there will be a period of time where your white blood cells are then going to be vulnerable to this antibody-dependent enhancement just because you haven't made enough antibodies yet, right? So continuing, recent advances in understanding of the receptor recognition and cell entry mechanisms of coronavirus have also allowed us to use coronaviruses as a model system for studying antibody-dependent enhancement. And that is, like, to me, that's not nice. It's not nice that we're going through a global pandemic with the coronavirus, knowing that coronaviruses are used as model viruses for antibody-dependent enhancement. That does not speak well to me. Continuing, in the study, we first demonstrated that the MERS-CoV receptor binding specific neutralizing monoclonal antibody binds to the RBD regions of the MERS-CoV spike, and further show that the monoclonal antibody mediates MERS-CoV pseudovirus entry into the Fc receptor expressing human cells. Moreover, a SARS-CoV receptor binding domain specific neutralizing monoclonal antibody mediates ADE of SARS-CoV pseudovirus entry. Wait, let me... Oh, they must have done... Okay, I think I might have skipped this, but apparently they did the same thing with SARS as well. Did they do that here? Okay, well, I'll have to read through it later, but yeah, apparently they said that for the original SARS, it mediates the ADE of the SARS-CoV pseudovirus entry as well. These results demonstrated that ADE of coronavirus is mediated by neutralizing that ADE of coronavirus is mediated by neutralizing monoclonal antibodies. That I think they meant to say sub-neutralizing, or the antibody itself is neutralizing, but maybe there's sub-neutralizing concentrations. In addition, the same coronavirus strains that led to the production of fully neutralizing monoclonal antibodies can be mediated to go through ADE by these neutralizing monoclonal antibodies. Our results differ from previously observed ADE of flavivirus, in which primary infections and secondary infections are caused by two different viral strains, in which ADE-mediated monoclonal antibodies are only sub-neutralizing. Okay, so okay, this makes sense. So they're saying that for the coronaviruses that they're studying, the antibodies that they're using are neutralizing antibodies. They are, so for instance, with dengue virus and the flaviviruses, the antibody-dependent enhancement was being used or being mediated by antibodies that were imperfect. They were imperfect at doing their job. But for the SARS, and the reason why they were imperfect was because they were working on a different strain of the dengue virus or the other flaviviruses. But they're saying here it doesn't need to be an antibody that is only sub-neutralizing. It could be an antibody that is absolutely perfect at stopping the virus. It could be an absolutely perfect antibody. But if the concentrations are too low, it's functioning like an imperfect antibody. So they're saying it's not just the imperfect antibodies you need to worry about. It's also the perfect antibodies that you produce as well. And so you got to make sure that those concentrations are high enough. So this could tell you that, especially if we're going to move into the realm of vaccine development, that that you might need a booster shot frequently, because if you have only low levels of the antibody, that might not be enough to fully protect you, and in fact, it could make you more vulnerable. And we do know that the amount of antibodies that you produce is somewhat correlated to how severe your disease was. So the more severe disease you had, at least for SARS-CoV-2, I should say, the amount of antibodies you produce is correlated to how severe the disease you had. So the more severe disease you have, the higher ramping of the antibodies you've made. So that could tell you that you are safer for longer than someone who just got a small amount of it and then ended up, getting it through their system very quickly. So similar to chickenpox for young kids, um, it might be they were able to get rid of it quickly, but then as they get older, as the immune system weakens and they have less circulating antibodies and less white blood cells that are able to ramp up production very quickly, um, then they could get shingles later, and it is a lot worse the second time around. So let's keep on going. So yeah, they're saying our results differ from those other studies that say that the antibody-dependent enhancement was done by the imperfect antibodies. Therefore, our study expands the concept of ADE of viral entry. So it's also not saying that coronaviruses aren't also using imperfect antibodies to do antibody-dependent enhancement. They're just saying that the perfect antibodies are also able to do that, which is deeply concerning for me. And so this puts people like me at a catch-22, right? So I've been describing some of my concerns where if we are not producing the right antibodies, if we're producing antibodies that are attacking parts of the spike protein that can do molecular mimicry of proteins and peptides that your own body produces, I will want those antibodies to lower or to lower in concentration very quickly after I've recovered, because those antibodies are going to be affecting my metabolism. So I do not want those to exist in high concentrations. But then we might be put into this catch-22 where if our antibodies get too low, then we end up mediating this antibody-dependent enhancement, right? So that might end up making the disease much worse. So what I would like to see is vaccine development that is very explicit in which part of the spike protein you end up making an antibody against. And by doing so, you are going to be subverting the autoimmune response, or you're going to be subverting your autoimmune attack of your own peptides. Okay, but what was I gonna follow? But we're going to need regular booster shots, is what it's sounding like. If we do decide to go down this route, we're gonna need regular booster shots, which I don't like. I don't like the idea of being subjected to needing these shots all the time. So what would be beneficial is knowing what parts of the spike protein you can develop antibodies against that do not mediate antibody-dependent enhancement. So if you could figure out which parts of the spike protein do not do this ADE, then that would be beneficial for you, because then you could make those antibodies that, one, do not cause autoimmune responses, and two, do not cause the antibody-dependent enhancement. And if you can get both of those, then you might just need one or two booster shots, but then your body will be able to not only fight off the virus, but then ramp up production when need be. So you won't always have to be at high levels of the antibody. You could end up producing less antibodies later, and then not worry about this ADE, because you know that your antibodies, or the antibodies that you are producing, are not participating in this. Now, is that possible? Do I know this happens? What would be one strategy that could cause that? So here, I think they've made mention that making an antibody that binds to the receptor binding domain while it's open, and that causes conformational changes in the spike protein, which uh, leads it to be more cleavable by proteolysis. Maybe an effective strategy would be to produce an antibody that forces the spike protein trimer to remain completely closed, right? So if you could find antibodies that sequester the spike protein and leave it in a completely closed state, maybe that is something that would confer some resistance to the white blood cells um, that end up becoming more susceptible to the virus. That is a possible strategy. In fact, I would prefer that strategy over an antibody that would work um, that works on the receptor binding domain while it's open. Granted, would those types of antibodies be as effective? Probably not. Um, Probably not, because it would probably only take a few mutations to render that antibody useless. But let's go with that. I mean, I just want to look at multiple multiple strategies here. Anyways, continuing, it looks like, oh gosh, what a long paper. I mean, that's good, but anyways, we examined the molecular mechanisms of ADE of coronavirus entry, and we showed that the oh wait, did I already read this? Oh yeah, no. We showed that the monoclonal antibody binds to the tip of the MERS-CoV spike trimer, where the RBD is located, and the monoclonal antibody binding likely stabilizes the receptor binding domain in the standing up position, triggers a conformational change of the MERS-CoV spike, and exposes the previously inaccessible S2' site to proteases. During the preparation of the manuscript, a newly published study demonstrated that SARS-CoV receptor binding specific monoclonal antibodies named S230 bound to the ACE2 binding region of SARS-CoV receptor binding domain, stabilizes the receptor binding domain in the standing up position, and triggered conformational changes of the SARS-CoV spike. In contrast, a MERS-CoV RBD specific MAB named LCA60 bound to the side of MERS-CoV RBD away from the binding region and stabilize the RBD in the lying down position. Okay, so this is I'm learning more every day. So this is the strategy that I was just suggesting earlier. Was let's possibly make antibodies that don't stabilize the open conformation. If we can make antibodies that stabilize the closed formation, that might make the virus a lot less infectious. And furthermore, it might be a mechanism that might protect us from antibody-dependent enhancement. Interesting. And did not trigger conformational changes of the MERS-CoV spike. Good. These published results are consistent with our results regarding MERSMAB1 triggered conformational changes of the MERS-CoV spike, together suggesting that in order to trigger conformational changes of the coronavirus spike, MABs need to bind the receptor binding region in their receptor binding domains and stabilize the receptor binding domain in the standing up position. Granted, there might be antibodies that don't bind the receptor binding domain and are able to bind close to it, which could theoretically push it up. But here they're just saying that it is necessary, although I could definitely see ways where that is not completely necessary. Moreover, our study revealed that ADE of MERS-CoV entry follows the same pathway as the DPP4 dependent MERS-CoV entry. Specifically, proprotein convertases partially activate MERS-CoV spike. So they're saying here that the mechanisms by which the coronavirus is able to infect the white blood cells is the same proteolytic methods as the normal binding to the DPP4, which for SARS and SARS-CoV-2, instead of DPP4, that would be ACE2 receptor. So the MERS-CoV spike can be further activated and fuse membranes on the cell surface. Otherwise, MERS-CoV enters endosome and lysosomes, where lysosomal protease activate MERS-CoV spike for membrane fusion. So here they're saying if the cell surface proteases are activated, then or present, then the MERS can fuse on the cell surface. But sometimes the viruses can be consumed by the cell and be brought into the endosomes and lysosomes, which is where they would normally be digested, except in this case, the digestion process can actually liberate the virus genetic material and then infect the cell later. So, as we said, multiple strategies to get into multiple cells. And then I should also mention that this might be a strategy for the viruses to make bioactive peptides. Now that I think about this, being sucked up into the lysosomes might actually be a strategy by which viruses might be able to be broken up into a bunch of different pieces, and then those different pieces then might play bioactive roles in the cell, or if the cell dies, then it might be playing a bioactive role in hormone changes, for instance. Interesting. Taken together, our results show that the RBD-specific neutralizing monoclonal antibodies bind to the same region on coronavirus spikes as viral receptors do, trigger conformational changes of the spikes as viral receptors do, and mediate ADE through the same pathways as viral receptor-dependent viral entry. In other words, RBD-specific neutralizing monoclonal antibodies mediate ADE of coronavirus entry by functionally mimicking viral receptors. So here they're saying that the antibody is functioning like a receptor. It is causing the same changes to the coronavirus spike proteins as a receptor does. Which goes to show you just how amazing these antibodies are at functionally mimicking a lot of the receptors that you have in your own body. Truly is remarkable. Finally, we analyze ADE of coronavirus entry at different antibody dosages. MERS-CoV entry into cells. Yeah, and this was these that I showed earlier. And demonstrates complex monoclonal antibody dosage-dependent patterns. As the concentrations of MAB increases, viral entry into the DPP4 expressing cells is inhibited more efficiently because the monoclonal antibody binds to the spike and blocks the entry. But the FC receptor expressing cells are at first enhanced. At first, they become more vulnerable, but as you increase the number of antibodies, eventually those antibodies are protective. So if you are making antibodies, you want to produce a lot of them to actually take care of the virus rather than just leading the virus to greener pastures. So let's see. If few of them normally percent. Okay. So expressing both DPP4 and FC receptors is first inhibited, then enhanced, and finally inhibited again. Oh, so yeah, if they express viral entry into cells expressing both of the receptor and both receptors, I should say, for both the antibodies or the straight up spike protein, then it is first inhibited, but then it's enhanced and finally inhibited again, which you can see here. First inhibited, then enhanced, inhibited again, and here it's first inhibited, then enhanced, and then inhibited again as you increase in concentration. There exists a balance between these two entry pathways that can be shifted and determined by the monoclonal antibody dosages. Importantly, ADE occurs only at intermediate MAB dosages. Our study explains an earlier observation that ADE of dengue virus occurs only at certain concentrations of monoclonal antibodies as well. So this is confirming the results of a different lab where they did the same experiments on, or similar experiments on dengue virus, and it also showed that for dengue virus, high levels of antibodies were protective, but low levels of antibodies were not. And so while many tissues express either DPP4 or the FC receptor, a few of them, most notably placenta, express both of them. So this could indicate to you that the placenta would be a very vulnerable organ for MERS infection for the second time around. So for other viruses that use viral receptors different from DPP4, there may also be human tissues where the viral receptor and FC receptor are both expressed. So for other viruses like the SARS and SARS-CoV-2 coronaviruses, they'd be expressing the ACE2 receptor instead of the DPP4, or I should say they'd be using the ACE2 receptor instead of the DPP4. The expression levels of these two receptors in specific tissue cells likely are determinants of MAB dosage in which ADE would occur in these tissues. Other determinants of ADE enabling MAB dosages may include the binding affinities of the monoclonal antibodies for the viral and FC receptors. Overall, our study suggests that ADE of viruses depends on antibody dosages, tissue specific expressions of viral and FC receptors, and some intrinsic features of the antibody. Excuse me. Something that sticks out to me as well here, and geez, 2 hours. Something that sticks out to me is antibodies are not evenly distributed throughout your body, right? So there might be some locations in your body that actually have lower levels of antibodies, would if you are immune to a particular virus, right? And you are producing neutralizing antibodies, those neutralizing antibodies are not going to be in high concentrations in your entire body. So there are locations called immunoprivileged sites in which you do not have as many white blood cells in those areas, and you do not have as many antibodies in those areas. So this is why I was a little concerned when we discovered that viral RNA for SARS-CoV-2 was present in semen, because the testes is known to be one of those immunoprivileged sites. So, and we also know that testicular tissue is very highly expressive of the ACE2 receptor. So what I'm worried about, what I will look into is the FC receptor also expressed on some of these testicular tissue cells? If so, that could indicate that the testes would be a prime location for both the ACE2 receptors to mediate entry into the cell, and then also the antibody levels would be low enough that the FC mediated entry might also be of some concern. Granted, since it's so immunoprivileged, it might be that as if a virus is able to get a hold in your testicular tissue, it's there for good, and you can't get rid of it. That is another concern. So, for instance, with Ebola, a lot of people who recovered from Ebola ended up having permanent eye damage months down the road, because eyes are an immunoprivileged site. So your immune system doesn't work upon your eyes as effectively as it does the rest of your body. And so some Ebola virus was able to get into the eyes of people, and once it was there, even after you've recovered, they could still be like doing their thing. So even after you've created neutralizing antibodies against them, the eyes still were, I guess, hosting the Ebola virus, and therefore damage to the eyes could have happened even after surviving. Okay, let's see. I'll just read through this really quick. So our findings not only reveal a novel molecular mechanism for ADE of coronaviruses, but also provide general guidance on viral vaccine design and antibody-based antiviral drug therapy. As we have shown here, RBD-specific neutralizing antibodies may mediate ADE viruses by mimicking the functions of viral receptors. Neutralizing monoclonal antibodies targeted other parts of viral spikes would be less likely to mediate ADE. if they do not, and just keep this also in mind, you do not make okay, well, it exited out. Don't worry about it. So you do not make monoclonal antibodies yourself. You will make polyclonal antibodies. You will make multiple antibodies for viruses. So what does this tell you? It is still possible that even if you do get infected with this virus, you're probably going to be producing a plethora of different antibodies that can attack this particular virus. So you might have some antibodies that are participating in the antibody-dependent enhancement, but maybe they are not allowing the structure to be in the open form, right? So maybe they're not fully going to be participating in ADE, but you just need one antibody that is binding to the receptor binding domain and keeping it in the open position, which would then expose the other proteins for proteases and therefore allow fusion of the virus to the cell. I'm just spitballing right now, but that would be a possible mechanism by which it could work. So, shoot, I accidentally stopped the recording, but let's see, I could just read a few more important sentences, but the in-glass systems used in the study provide a model framework for ADE, and future research using in vivo systems is needed to further confirm these results. Yeah, it's kind of weird that they're talking about doing research in vivo systems for this antibody-dependent enhancement right as the coronavirus ended up becoming human-born. Regardless, our previous study shows that a humanized version of MERS-MAB1 efficiently protected human DPP4 transgenic mice from live MERS-CoV challenges. So, suggesting that given the antibody dosages used in this previous study, as well as the binding affinities of the MAB for human DPP, the receptor-dependent pathway of MERS-CoV entry dominated over ADE in vivo. So in vivo, the receptor-dependent pathway dominated over the antibody-dependent enhancement, in vivo at least. Thus, future in vivo studies may need to screen for a wide range of antibody dosages and also for a variety of tissues with different ratios of the DPP4 and the Fc receptor. And although ADE has not been observed in MERS-CoV in vivo, our study suggests that ADE occurs under some specific conditions in vivo, depending on the antibody dosage, binding affinity of the monoclonal antibody for DPP4, and tissue expression of DPP4 and Fc receptor. Moreover, the mechanism that we have identified for ADE of MERS-CoV in vitro may account for the ADE observed in vivo for other coronaviruses such as SARS-CoV and feline coronaviruses. Overall, our study reveals complex roles of antibodies in viral entry and can guide future vaccine design and antibody-based drug therapy. So with that, I'm going to X out, and thanks for listening. And if you have any questions, leave them in the comments down below. And what else do I want to say? To me, I just want to bring up that it is very concerning not knowing whether or not the mild cases of the SARS coronavirus are actually conferring protective immunity and giving you a protective amount of neutralizing antibodies. That to me is concerning that we can't figure that out. For instance, there are some children that are showing signs of the pediatric multi-inflammatory syndrome or multi-system inflammatory syndrome, and in children, so MIS-C. Some of them don't even test positive for antigen. So what is that? Or not positive for antigen, they don't test positive for antibodies. So what does that tell me? It tells me that in many cases it sounds like your antibodies response wants to be suppressed in some way. The body does not want to just be producing antibodies willy-nilly against this virus. And why would that? Well, if viruses are mimicking human antigens, then the body might be unable to make antibodies because the body frequently kills cells, or I should say, in the maturation process of white blood cells, if they are, I don't remember which type of white blood cell, but they will, the ones that are producing these antibodies, if they end up producing antibodies that attack human proteins, they end up getting exterminated. Normally, this is through the normal maturation process. Granted, and because of that, if a virus is expressing a lot of antigens that look similar to humans, it'll be very difficult to find the white blood cell that has an antibody that will bind to the virus, but also be different enough from humans, right? Now, but where was I going with this? So, to me, I don't know if a lot of the serious cases that we're seeing are the serious cases actually cases where someone actually got infected twice, like it was their second infection, which could theoretically happen if the first sweep through was just a mild disease, and then a few weeks later you got another inoculation, and that second time around was actually much more multi-systematic. Possibly, I don't know, because we're not getting good data, or we're not getting enough good data for this, unfortunately. And we keep on going around saying that let's just give immunity passports to people, and to me that's scary. We are not treating our enemies with respect. We're not treating our enemies with respect, which is the way that you defeat an enemy, is to size it up properly. And I do not believe that we size it up properly. I do not believe that the media size it up properly. And yeah, it's just a lot of it is disheartening. So hopefully science is able to catch up, and thanks to a lot of the really a lot of social distancing that we have been doing has been remarkable. I would not have believed, if you told me 2 months ago, that we would have basically shut down a huge chunk of our economy, and that we as a people decided to start wearing a lot more face masks and started to distance ourselves from each other. I would have not believed you. So I'm actually very, very happy to see that we have come this far in protecting each other and allowing science to catch up, which it looks like it is. It is catching up, at least for now. And I really, really hope that it can remain caught up, because there's a lot of disheartening things that I hear right now as far as what is being spread on the news, and it's I don't think it's doing a good job at. I don't believe the media is doing a good job at giving people informed consent of the dangers of this virus and what is an acceptable level of exposure. So anyways, with that, I'm going to hang up, but thanks for listening, and I will see you tomorrow.